I had the pleasure of presenting in an educational session called Expanding Synthetic Lethality Strategies Beyond PARP. And the task that I was given was to discuss how we're now going beyond immunotherapy for the treatment of MSI tumors through the development of Werner helicase inhibitors. So the background to all of this is that we know that immune checkpoint inhibitors are the standard of care now for metastatic MSI high or dMMR cancers. And the FDA has indeed approved this based on the phase 3 checkmate 8HW trial, which looked at nivolumab and ipilimumab in this subset of patients with dMMR and MSI high metastatic colorectal cancer. And that particular trial showed that ipi-nivo demonstrated clinically meaningful and statistically significant improvements in PFS versus nivolumab monotherapy alone. However, the challenge is that not all MSI high and dMMR tumors respond to immune checkpoint inhibitors, and approximately 50% of these MSI high or dMMR tumors actually exhibit primary and acquired resistance to immune checkpoint inhibitors. So this is why Werner helicase inhibitors are so important to really provide an alternative to uh, the management of these patients in the resistant setting. And so the background to this is that when a helicase is a helicase enzyme that belongs to the RECQ family, and it has a critical role in the maintenance of genome integrity. And so when a helicase serves as a DNA caretaker enzyme and has an important role in unwinding non-canonical secondary DNA structures that are formed in response to DNA damage or during the DNA repair process. And so loss of Werner helicase actually leads to an accumulation of DNA damage during the cell replication in tumor cells with MSI that leads to cell death. And therefore, through synthetic lethality, Werner helicase inhibition in cancers with microsatellite instability have now emerged as a promising approach to target these MSI and dMMR cancers that are insensitive to standard of care immune checkpoint inhibitors. And these agents have been shown to lead to mitotic cleavage, to DNA double strand breaks and mitotic catastrophe, and eventually cell death. And it's very exciting that the timeline from the actual discovery of the Werner helicase synthetic lethality and MMR deficient cells has been very brisk. This was only discovered back in 2019, and over the years, through improved mechanistic understanding of this class of agents, improvements in innovative chemistry, and also culminating in clinical proof of concept and increased insights in the clinic with the presentation of early first-in-human phase 1 trial data just last year. And now there are currently multiple different Werner helicase inhibitor trials that are ongoing, certainly in the US and many more overseas. But at the last count, there were approximately 7 different Werner helicase inhibitor phase 1 clinical trials that are looking at this class of agents either as monotherapy or in combinations with either other immune checkpoint inhibitors such as ipi-nivo or pembrolizumab, and also standard of care treatments such as chemotherapy and also bevacizumab. Now, the whole importance of this class of agents is that they induce tumor regression in preclinical MSI, CDX, and PDX models, but there are no effects in MSI models with either Werner helicase, C727A mutations, so these are resistant lines, and also importantly, no effect in MSS tumors, and therefore that provides a very wide therapeutic index. And so in my talk, I covered the clinical data that are currently out there based on data from two different clinical trials. One was from Roche Vividion, currently now with Bayer. The second uh, clinical trial that I touched on was from Novartis. And both of these clinical trials essentially provided early clinical proof of concept for drugging Werner helicase in MSI tumors. It was shown that Werner helicase was safe and well tolerated when inhibited, with mainly low grade, manageable GI toxicities with these Werner helicase inhibitors. And importantly, there really wasn't any myelosuppression observed that we often see with DNA damage response inhibitors. Importantly, we observe responses in IO exposed and IO resistant patients with MSI tumors. And overall, whilst there was a modest overall response rate of approximately 5.5 to 14% between the two different trials, there was a high and impressive disease control rate of approximately 65 to 67% in these very resistant patients, including resistance to immune checkpoint inhibitors. And so we observed durable, stable disease and tumor regressions with both of the drugs from Bayer and also from Novartis. And overall, the patient benefit was also supported by metabolic responses as shown by FDG PET scans and also molecular responses as demonstrated by ctDNA studies on both of their trials. In terms of the future development, what we're now focused on are really how we can improve our understanding of the predictive biomarkers of response and also in deepening our understanding of resistance to these Werner helicase inhibitors. Very excitingly, different groups, such as the one from the Sanger Institute, from Matthew Garnett's lab, Gabriel Pico has shown that using long read whole genome sequencing, higher TA repeat burdens is actually associated with greater sensitivity to Werner helicase inhibitors, supporting their potential use as predictive biomarker assays of response. In terms of resistance, it's also been shown that on-target mutations do confer resistance to Werner helicase inhibitors in MSI cancer cells. And interestingly, some resistant mutants actually remain sensitive to alternative Werner helicase inhibitors with distinct mechanisms of action, suggesting that resistance may actually be compound specific and potentially therapeutically tractable. In my talk, I also discuss, different questions that currently remain and ones that we should now focus on with the many different Werner helicase inhibitors that are currently out there. For example, number one, what is the optimal setting for these Werner helicase inhibitors given the great response that we already see with different immunotherapeutic agents? And should we expect cross-resistance between immunotherapeutic agents and Werner helicase inhibitors, especially given the very distinct mechanisms of action between IO agents and a targeted therapy like Werner helicase inhibitors that really acts through synthetic lethality. In addition to that, it's also curious to examine if the Werner helicase inhibition leads to an impact on the immune system as well, and also to investigate why we do see a disconnect between the overall response rate and durable disease stabilization and regressions that we do see with different Werner helicase inhibitors. And ultimately, it's important to examine if durable stable disease and regressions will ultimately lead to improved PFS and overall survival for drug approval. In addition to that, in clinical trials, we're now looking at different combinations, certainly immune checkpoint inhibitor combinations, for example, with pembrolizumab, and in the future with ipi-nivo, and also looking at combinations with chemotherapy agents and also with bevacizumab. Key question as well is, can we go beyond just MSI tumors, for example? Should we be looking at TA repeats as a predictive biomarker of response? And also, is there context dependency between different cancer types or can this be a truly tumor agnostic strategy using a predictive biomarker? And so, the key takeaways from my talk was that when a helicase is a helicase enzyme that plays a critical role in the maintenance of genome integrity, and loss of when a helicase results in an accumulation of DNA damage during cell replication and tumor cells with MSI that leads to cell death. And I also discussed that when a helicase inhibitors are indeed a promising approach to target MSI cancers that are insensitive to standard of care treatment. Thank you very much.